just I want to, before starting, just to thank Duncan and Sofian for the invitation. That's my first time to this meeting, and I will present the data we have on the L-type channelopathies that causes autism. And in the last several years, we found evidence that there are some common gating changes among the different L-type channel which causes this form of pathologies. Before I go on, let me just remind you about the L-type channels. The L-type channel plays a key role on heart and smooth muscle contraction, neuronal development and maturation, long-term synaptic plasticity, sensory transduction, and hormone release. And among the milestones we had in the literature, we had the cloning of the cardiac CACNA1C in 1989 by the group of NUMAS, and then the cloning of the neuronal L-type channel in 1991 by Tersnach group. And in 2000, we had the first knockout mouse, which was reported as lethal mouse. So that means that L-type channels, the CAV1-2 channels, are really tremendously important for life. It was at the time surprising that there was no reports of channelopathies in this field, and it was surprising because if it's so lethal, why we don't find any humans having some problems? Well, the first report came exactly in 2004 when the marketing group reported the mutation, a glycine 406 into an arginine at the end of the S6 segment in the first domain. That was a mutation in exon 8A, which is an alternative exon. It's an alternative exon cloning for this S6 segments. The report was that people had lethal cardiac arrhythmias, congenital heart disease, syndactyly, they had immune depression, they had also intermittent hypoglycemia and autism. A year later, the same group came with the same mutation, but on the other exon coding for the S6 is exon 8, and they had the same mutation G406R, which causes autism, and another mutation G402S, which doesn't cause a mutation but causes heart problems. The mutation causes a delay in the inactivation of the channel. The channel is less inactivated, carries more calcium into the cells get sick and start having problem with the heart beating. There is a long QT, the QT is around doubled, and there is a polymorphic ventricular tachycardia, which is very serious for these patients. After this report, there was many reports reporting mutation in different positions, but as you see clearly, you see the position is always in one of the S6 helix, which means that they have the same effect on autism. So what changes the gating makes the autism. Of course, there were also many other mutations. All these ones causes only heart problems to these patients. Well, the problem was discussed in many cell preparation, heterologous X cells, iPSCs used, but never on native cells. One of the nice reports that we liked and we could repeat in Torino was that the G406R mutation shifted the activation of the L-type channel to the left and cause autism. If we study the other mutation, the G402S mutation, causes no autism. So we focus on this property of the calcium channels, of the CAV1-2. And, but before doing, deciding where to go, we got this report, and it was sent to me by Alexandra Pinguera in Innsbruck. One day she sent me this paper and said, look, Emilio, look what's happened at these patients when they look at the autopsy of this TS1 young female. She died at 2 months, and they reported that the medulla, the adrenal medulla, was completely destroyed, collapsed, while the cortex of the adrenal gland was maintained pretty much in a good position, in a good shape. The report was that this chromaffin cell, these glands lost a lot of chromaffin cells because of the abnormal calcium entering into the cell. They could cause, a downregulation of mitochondrial function. After that, we then decided, before to go to neuron and see what neuron do, to see what in chromaffin cell does the mutation, just as a model, because we knew very well. And I tell you in few slides what chromaffin cell does. Chromaffin cell are controlled by the splanchnic nerve, which discharges train of action potential, release acetylcholine. The terminal release acetylcholine through the nicotinic receptor. There is a strong depolarization, firing of the cell, which of course respond very nice, and release adrenaline. But we found recently, and this is very important that you know, because otherwise you don't understand all the rest of my talk, we found that without innervating the cell, taking the cell in culture, they regularly find. They have a tonic firing, regular tonic, burst firing, with the less percent this one, but this is a very important one, because if this happens, we have for half a second the entry of calcium, then we have a lot of release of adrenaline. We were questioned about this story and saying that, that could have been an artifact of the recording using the patch pipette. And then we repeated the measurements on MEA, and you see that the chromaffin cell attaches very well. And we in this case, we just attach the cell and nothing else, and we have a nice recording of action potential, this one here, and then we have also intermittent burst firing, rather different from the one I just reported. We are finishing the things and publish. And the good things, which is very important, is that each recording is not an action potential, but is a current. You see, this is a sodium-calcium current, this is a sodium-potassium current, which is the current we measure here times the resistance. Okay, this is another story. The chromaffin cell fire like a neuron, so it's very nice to study because they express sodium channel NaV1.3, NaV1.7, particularly NaV1.3, all the calcium channels. They express also lots of PK and SK channel coupled to the calcium channel. They have all the KV channels. They have the KV7, KV1.3, K4, KV11, and then they have even TASC. And in the list there are not the TRIP channel, which are coming out soon, because we have also evidence for the TRIP channels. Well, let's go into this L-type channel. The L-type channel can be measured by every chromaffin cell. They have large currents. These are calcium current. They are not L. These are calcium current. But if we separate, by blocking with nifedipine, most of the calcium current, 0.5% is L. And this is nice to study a mutation on chromaffin cell, because there are a lot of L-type channels. Particularly, we have half L-type channel which are CAV1.2 and half of the channel which are CAV1.3. And this we published recently. Well, given this, the point was, what shall we do? Shall we go and look for injecting mutated clones in the neuron? I met at a neuroscience meeting Randy Rasmussen and Glenn Abbott, who just at the time were published this paper showing that there is a mouse model they prepared a mouse model with a mutation, with the G406 mutation, G406R mutation, but in the homozygous mouse, it was lethal. They had to make an heterologous and put a cassette, a myosin cassette, a neomycin cassette on the exon 8A in order that the exon 8 could not be, too much expressed. We got the mouse, we were very happy, and we started looking at the first how much exon 8 and exon 8A were expressed. In the norm and the wild type there was much less than we expected, exon 8, but there was much more because of the neocassette in the exon 8A. So we estimate that we had enough probably mutated CAV1-2 channel in order to make the measurements and to make the experiment we wanted. And then we started. So we started with measuring the calcium currents. These are L-type calcium currents isolated pharmacologically. We can block very nicely with nifedipine. As you cannot identify here, but you see the IV is slightly shifted to the left, which is very important. The conductance is the same. And more important, the density, the current was not much changed, but if we divide by the capacitance, since the capacitance became bigger, you cannot believe, but if you're not an electrophysiology, you don't know that if you measure very nicely the capacitance, you measure nicely the surface of the cell. So you know exactly how the surface is. That means this suggested that the cell swells a little bit, and so if we divide the two parameters, we get a decrease in the calcium current. But the decrease in calcium current, I will show you now what's happened. We tested whether the inactivation was slowed, and it was slowed here, and it was calcium independent, was voltage dependent because we found it with barium, and we found it with calcium, but the calcium dependent inactivation around here was not changed. Then we measured the steady state inactivation, which was shifted again, and as we published recently, if we multiply the steady state inactivation by the conductance here, we get what we call the window current, the window calcium, which is very important because the window calcium current, it's a small, tiny current, a few picoamps, but it passes forever, for 24 hours a day. So it can be little in the sense of the total current, but it's continuously passing, so it makes a lot of change into the cell. And you see here, we multiply the steady state by the conductance and then for the driving force, and then we get the window current, and then you see the shift to the left. This made a big change to the firing. The big change to the firing was that the resting potential was not altered. We get a change in the ratio of tonic to burst. You see, it was large for wild type and low for large for wild type, lower for the wild type. But not only the number of cells, we had also a change in the action potential shape. So here are the train of action potential. Here you see that the amplitude decreases, but also changes the shape. And to see the shape, you know, electrophysiology looks always at the phase plane plot, and you see very well here that there is a decrease in the first negative derivative here, you see, while it changes also the amplitude around here. Well, this means, and this was for the tonic, for the busting cell was practically the same. So we got the impression that there was a decrease in sodium channels. We measured the sodium channel and we found really that the sodium channel were less expressed in TS2 neo. And of course, again, the current was not significantly different, but again, with the increase in the capacitance, the density of the channel decreased. And we tested the IV, was not changed, the inactivation was not changed, was a little bit changed the steady state inactivation, which means that if we have the cell around -40, we get a loss of currents also here. Because of this, I wanted to know why we had such a bad recording in in the action potential, but also looking at the cell. The cell were really very different between wild type and TS2 neo. There was an area of vacuolinization here, all this white spot here, and if you look and compare the difference, the mitochondria and the wild type are very well resolved, but are very badly resolved. So the cells is really in a bad shape, and of course, my colleagues in Torino, they measure the area, and the change in the area is exactly the change in the capacitance we had. So that was really rewarding. And then finally we thought, okay, if there is no way, let's start at least to look whether a good blocker of electric channel can recover the cells. They are not neurons, they are chromatin cells, but let's see. And we started incubating the cell, the TS2 neo cell, with nifedipine, which is the most used calcium alpha channel blockers. And of course, we started with high concentration, 3 micromoles, 10 micromoles, and the cell died completely. So we lowered, we found that 0.1 was okay, but 0.3 was fine. So we could recover fully the nice amplitude of the cell by just blocking the flux of calcium into the cells. And of course, we could recover the percentage of cell which has a tonic firing with the bar. This is the blue. We could recover the shape of the action potential. You see here the amplitude that becomes bigger. Here we recover the half width, the anti-peak, and here, and here also we could also recover the capacitance change. The secretion change, because in chromatin cell we have the advantage that with carbon fiber we can record directly the release of catecholamines. Good. After that, we left the chromatin cell and started with neurons. So in neurons, we found more or less the same thing. So we found that between wild type and mutated neurons, there was a 25% decrease in the cell, and particularly there was a decrease in the glutamatergic with respect to the GABAergic response. In the neurons, we could prove exactly as we worked on chromatin cell that the mutation is there and the inactivation of the L-type channel decreases. Here's the percentage of currents here increasing. We can show that there is a shift in the calcium currents. This is very tough to measure in hippocampal neuron because they have dendrites. So we made the measurement at very few days in vitro. The shift in the IV is the same shift in the conductance. And of course, we had the same more or less property on the steady state inactivation and the same property on the window current, on the window calcium current, which means the cells are sick like the chromatin cell, and one of the reasons why we get autism. We wanted to go further, and we started measuring the GABAergic potency, the strength of the GABA. So we use what we have in our lab, the monosynaptic hippocampal neuronal network. It's a very nice prep. We can stimulate presynaptically and record postsynaptically. And you see that if we block pharmacologically the glutamatergic neurons, we have, you see, control and wash, control, nice recording, pyrrhotoxin blocks everything, and then we can wash it again. So this is typically GABAergic. And then very simple, we get an increase, a tremendous increase in the postsynaptic currents. Here is. To see whether there is an increase in the receptor or in the probability of release, you just have to run a pair pulse depression protocol, and we found that the pair pulse protocol increases the pair pulse depression, which means there is an increase in the release. We stopped here because we have now to collect money to proceed, but that was, these are unpublished data, of course. Let me show you then, and I finish in a few minutes, what's happened to the other, to the close parents of the CAV1-2, the CAV1-3. CAV1-3 channel is very important. The CAV1-3 channels, it's expressed in the heart, in the brain, endocrine cells, particularly in the adrenal medulla. We had several papers on this with the group that works at that. I will mention now. Important is that deletion of CAV1-3 in humans causes deafness and bradycardia, which that means plays a role, and missense mutation causes severe multi-organ syndrome. If you look here, I borrowed this slide from my friend Jörg Strisnig. You see there are reports on mutation more or less similar to the Timothy syndrome, which makes the same change in the currents and has more or less the same effect. Autism, spectrum disorder, intellectual disability. There are other mutations causing, for instance, primary aldosteronism, congenital hyperinsulinism, but particularly there is one here, this one, at the second, the domain 2 S6 segment here, the A746G, which is a mutation in a patient who had autism, but no serious other problem, because the CAV1-3 is in the brain, it's in the heart, but not so tricky, not so important like the CAV1-2. Jörg Strisnig, with whom we collaborate since 20 years with Nadine Orner, they organized to have the heterozygous CAV1-3 AG mouse model with this mutation incorporated. The mouse has been now, we send all this data now to for publication. They have developmental delay, social deficit, hyperactivity, no major changes in neuroanatomy. So we tested that, and then I show you the same things we found on TS2, but with different, of course, results. Here again, chromaffin cells are fantastic to patch. I tell you, it's very easy. You get, they are spherical. You don't have any patch clamp problem. So it's really a paradise for the electrophysiologist. Here are the currents in wild type and knock-in mouse. Again, we see the shift. So this is a constant. We had the shift in the conductor as well. And different from the TS2, we had perfectly the same currents. There is almost no difference because these two mean values are the same. There is a change in the inactivation, but it's different. The inactivation is not delayed, but is accelerated. You see here. There is a shift in the steady state inactivation. There is the same shift in the in the window current, very nice, which means that here again at rest we have a current pumping calcium in the cell and changing the characteristic, the property of the cells. With Laura, she's now in the States. With Laura, we checked the action potential story. First of all, it's different from the TS2, but this is different because CAV1-3 and CAV1-2, they are coupled to the BK channel in a very different way. So we had increased in the resting potential from around -48 to -55. We had again the change in the percentage of cell from tonic to silent and from firing and silent. There were more silent cell than firing. And when we tested the resting potential, the activity, we found that there was a strong decrease in the frequency. Not much change in the action potential, but basically on the interspikes interval. I think I finish here. This is the same, I'm sorry, this is a rest, but we can induce the firing by just applying a current 4, 10, 16 picoamps, and we see that we had a very fast initial frequency, but very delayed steady state frequency. And if we plot the value of the initial frequency, there is not much change between wild type and lock-in, but there is quite a change in the steady state, which is exactly in line with what we found at rest. Exactly, you see here, more expanded, the initial is equal, and the late is a little bit slower. So I think I concluded. I'm sorry. So this is just to say that CAV1-2 mutation and CAV1-3 mutation, and they are different channels. They have the same hallmarks in the gating. So they shift both to the left. The inactivation is not important in neurons and chromaffin cell. So we can conclude that TS2 and CAV1G mutation causing autism induce a similar leftward shift of channel activation and steady state inactivation with consequent increase in window corner. I think you can agree. Both mutation alters markedly the MCC firing property, although through different molecular pathway, and this depends on how the LTAC channel are coupled with the BK channels. The G406R mutation potentiates GABA synapses, which is the future work we have to do, and likely alters the excitation inhibition balance of hippocampal neurons that we have to, of course, to expand. And of course, overnight incubation with the GHP rescues the normal CC excitability. But the message I want to send to everybody is that more than using a blocker, in my view, we have to find a compound which shift back the activation to the right. Difficult to do. I mean, I discussed with all my calcium channel colleagues, everybody, but most now agree that we have to look for a kind of molecule that could be good for some types of autism, at least. And finally, let me just remind you that mouse chromaffin cells are an excellent model system for studying channelopathies and neurodegenerative disease. There is long papers in comparative physiology. These are all the people worked. I show the picture of all of them, so I don't have to repeat. Thanks for the attention.
Thank you very much, Emilio. And we're running a little bit over, but time for a question at the back. Yes, please. Right at the back, please.
Again, Alex from Midterm. Thanks, Emilio. It was a very nice story. So I have two questions. The first one is because you just mentioned the difference in the two mutations you showed.
Speak louder.
Sorry. You just showed the two different examples of the two different mutations, 1.2, 1.3. They affect the firing of the chromaffin cell differently, and you said it's mainly how they are coupled to the... sorry? Can you hear me now?
Yeah.
Okay.
Better.
So basically you said the difference is mostly due to how they are coupled to the BK channels. But don't you think that the faster inactivation of the AG might also somehow contribute to the difference in the firing?
Today?
The difference in the firing?
Sure.
Yeah.
No, definitely. I'm sorry, I was.
Yeah.
This is the first question?
The first question.
Well, you are right. The two mutations make a shift to the left. CAV1-2 probably, I could not say anything, could probably be linked to the gene, to the gene expression, and change completely the activation, the expression of the cells. It changes the mitochondria, it changes the gene, and then we get less sodium channel and things like this. The CAV1-3 mutation, CAV1-3 in chromaffin cell, maybe you remember or not, I did some seminar in Innsbruck. CAV1-3 is tremendously coupled to BK. That means if you block CAV1-3, you block also BK, and you get exactly the opposite of what you think. The fast inactivation works, of course, and my explanation for this is that at rest, calcium channel activates a little bit a more negative potential. That's why you go down. And since during the firing you have less entry of calcium and more BK is available, then you have a slow firing modes. This is my explanation, of course. We have to now to confirm in neurons. I mean, these are chromaffin cells.
Yeah, no, makes sense. Thanks. And the second question
Just
would,
very briefly, please.
yeah, would just be, did you, because you did overnight incubation with the nifedipine, did you also see an acute effect? You tried overnight incubation
Sure.
with nifedipine. Did you also see an acute effect?
Acute, you block the L-type channels. But good that you ask, because I didn't say the most important thing. If we give nifedipine 0.3 micromolar to the sick cells, we recover. But if we give 0.3 micromolar nifedipine on the wild type, we kill the cell. Okay. Which means, what is the message? The message is that the cell needs some leak of calcium during the resting state. If you block it, the cell doesn't work anymore. You understand? So that we had to do. Because one of the referees asked, well, you have tested the nifedipine on the TS2 mutated mouse. You have to prove also on the wild type, and we did it, but the cell dies.
Thanks.
Thank you very much, Emilio.
Okay, sorry.
All right. So